Hello, this is Rebecca Noel, and I am performing the last experiment for Dr. Pushpa's Chemistry 2 course called Chromatography of Food Dyes. So in summary, this experiment will allow us to learn how mixtures of compounds can be separated and determine what food dyes are found in certain foods by using paper chromatography. We will also learn how solubility is affected by polarity. We are given a 100 milliliter glass beaker, distilled water, salt, ruler, stapler, pencil, toothpicks, 2 packs of Kool-Aid, 1 pack of M&Ms, a set of food coloring, petri dish, well plate, and experiment bag. So our lab bag contains many food dyes: blue dye number 1, blue dye number 2, red dye number 3, red dye number 40, yellow dye number 5, yellow dye number 6, and an unknown. Each of these dyes will be placed into a petri dish, and then we will also put M&Ms in some of them. So the separation of the mixture is based on polarity. Most dyes are polar and will be soluble in water. We will absorb the dyes on paper, which is also polar. Then we will use slightly salted water as our solvent to move the dyes on the paper. Since some of the dyes are more polar than others, they are absorbed more tightly to the paper and are moved more slowly by the salt water. So FD&C dyes refer to specific dyes approved for human consumption under the U.S. government's Food, Drugs, and Cosmetics Act. Food coloring typically found in grocery stores may vary from brand to brand. One brand of food coloring, like the colors used for different food products, may consist of only one FD&C dye, while another brand may be a mixture of 2 or more dyes. So this experiment will show us the various color constituents of Kool-Aid, our grocery store's brand of food coloring, plus the other food items you select to test. So we will get 2 sheets of filter paper and draw a thin horizontal line with a pencil across the paper, about 1 centimeter from the bottom. And the line should be just barely visible. And with a pencil, we draw 9 small cross lines along the horizontal line, as shown. And the reason we do this is to test the travel of the dye up the sheet. So for each dye on the sheet to be tested, we A, first place a few drops in a well of the 24 well plate. B, dip a clean toothpick tip into the dye and then briefly touch the toothpick tip to the center of the appropriately marked section on the horizontal line of the filter paper. And C, this is a note, with a small drop of water on a little bit of Kool-Aid powder will be sufficient to make a sample to spot the paper. Small drop of water on the coating of an M&M will dissolve enough dye for the toothpick to pick up. The coating of blue and brown M&Ms is made up of very interesting dye combinations. And the second note, apply only a small drop of each dye on the paper. Big drops may spread over a greater area and overlap with other dyes. This is called band broadening. Although a larger dot will produce a more vivid color, a smaller dot will show the most distinct break between colors. So once the paper is spotted with all the dyes, we allow the spots to dry for a few minutes. And once we have all our calculations in place, what we will do is calculate the RF value. One important thing to remember is that We have to make sure not to touch the Petri dish sides. The solvent front will travel up the paper rapidly at first and then will slow down. So we have to let the solvent rise for a few minutes, but immediately remove the cylinder if any column moves up higher than 2 centimeters from the top of the paper. If all solvent is soaked up before the front has time to move toward the top of the paper, we will carefully add a little more solvent to the Petri dish. And record it all. Calculate the RF value. And that is the measure of the dye distance, the distance it travels, over the solvent distance. Okay, thank you very much.